walked into my sister's engagement party carrying research for Phase 3 cancer trials, wearing the same lab coat I'd worn for 3 years developing breakthrough treatments. But when I approached the family table, my Aunt Patricia announced loudly, "There's Dr. Rachel, still playing scientist instead of getting a real life. You're embarrassing, Emma, so please leave." 2 days later, Pharmaceutical CEO Jonathan Sterling introduced me to his stunned boardroom. "Gentlemen, meet Dr. Rachel Kim, our youngest." Before we jump back in, tell us where you're tuning in from, and if this story touches you, make sure you're subscribed. Because tomorrow, I've saved something extra special for you. Board director, whose research just saved us $2 billion. My name is Dr. Rachel Kim, and if you've ever been told your passion was just a phase, that your dedication was embarrassing to your family, this story is for you. Growing up in Palo Alto, I was surrounded by Silicon Valley success stories, but in my family, achievement was measured by traditional markers, stable careers, social status, and maintaining appearances that showed everyone you were living a normal life. My father, James Kim, was a successful software engineer at Google. My mother, Susan, worked as a financial advisor and believed that happiness came from work-life balance, social connections, and building a family. They had created a comfortable life in the South Bay, complete with weekend barbecues, family vacations, and dinner parties, where success was measured by how well-rounded and socially adjusted you appeared. But it was my grandfather, Dr. Henry Doc Kim, who understood the calling of science. Every Saturday, while other kids went to soccer practice or piano lessons, I spent my time in his home laboratory, listening to his stories about developing antibiotics during the Korean War. Grandpa Doc had been a field medic who later became a biochemist, and his converted garage was filled with microscopes, petri dishes, and journals documenting his research on infectious diseases. "Rachel," he'd say, adjusting the focus on an old microscope as we examined bacteria cultures, "medicine isn't about glory or money. It's about seeing problems that others can't see, and having the patience to solve them one experiment at a time." He handed me a small research journal bound in leather, filled with his methodical notes about compound interactions and treatment protocols. That journal became my roadmap. While my classmates dreamed of social media fame or startup fortunes, I dreamed of clinical trials and peer-reviewed publications. I threw myself into everything that would prepare me for medical research: advanced chemistry, molecular biology, statistics, and biochemistry. I studied disease mechanisms until I could recite the pathways of cancer metastasis and predict drug interactions. When I announced my intention to pursue a PhD in pharmaceutical sciences instead of medical school, the family reaction was swift and concerned. "Rachel," my mother said, looking genuinely worried, "research is so isolating. You'll spend your whole life in labs instead of helping real people. Why not become a doctor and have a practice? You could have normal hours and actually interact with patients." Dad was more direct. "Rachel, pharmaceutical research is incredibly risky. Most drugs never make it to market. You could spend decades working on treatments that never help anyone. What about job security? What about having time for a family?" But Grandpa Doc believed in me. He spent hours teaching me proper research methodology, showing me how to design controlled experiments, and connecting me with professors who could guide my graduate studies. When I received my acceptance letter to Stanford's Pharmaceutical Sciences program, he was the first person I called. "I knew you had the mind of a scientist," he said, and I could hear the pride in his voice. My family's reaction was different. They attended my PhD graduation, but every conversation included questions about when I would transition to something more practical and start focusing on building a personal life. At Stanford, I found my purpose. The rigorous research protocols, the collaborative problem-solving, the possibility of developing treatments that could save lives, it all made perfect sense. I specialized in oncology drug development, focusing on targeted cancer therapies that could attack tumors without destroying healthy tissue. My first major breakthrough came during my postdoc position at Biogenesis Labs, when I discovered something that would revolutionize how we approach cancer treatment. While working on routine protein analysis for Dr. Margaret Chen's team, I noticed an anomaly that everyone else seemed to be dismissing as experimental error. The anomaly appeared in cancer cell cultures that were supposed to be dying from our experimental drug compound. Instead of dying uniformly, about 15% of the cells were developing resistance through a mechanism that nobody had documented before. Most researchers would have adjusted the experimental conditions to eliminate this noise, but I saw it as the key to understanding why cancer treatments eventually fail. I spent 6 months in the lab, often working until 3 a.m., mapping the exact molecular pathway these resistant cells were using to survive. What I discovered changed everything. Cancer cells weren't just randomly mutating to survive treatment. They were following predictable patterns based on their genetic signatures. This insight led me to develop a diagnostic protocol that could predict which patients would develop resistance to specific drugs before treatment even began. Instead of giving all patients the same chemotherapy and hoping for the best, doctors could customize treatment plans based on each tumor's genetic profile. Dr. Chen reviewed my findings with obvious excitement. "Rachel, this is paradigm shifting," she said, studying my data spread across 3 computer monitors. "This could eliminate treatment failure for 60% of cancer patients. We need to get this into clinical trials immediately." My protocol worked exactly as predicted. In our initial study of 200 patients, customized treatment plans, based on my resistance predictions, increased survival rates by 40% compared to standard chemotherapy protocols. Oncologists from major cancer centers began requesting access to our diagnostic system. But the real validation came when I identified a completely new approach to treating pancreatic cancer, one of the most aggressive and treatment-resistant forms of the disease. Using my resistance pathway analysis, I discovered that pancreatic tumors could be made vulnerable by combining 3 existing drugs in a specific sequence that nobody had ever attempted. The breakthrough happened during a late-night experiment when I was testing drug combinations that conventional wisdom said would be toxic to healthy cells. Instead, I found that the sequence actually protected normal tissue while making cancer cells destroy themselves through their own resistance mechanisms. Within 8 months, my pancreatic cancer protocol was being tested at 15 major medical centers. The preliminary results were staggering. Patients with terminal diagnoses were achieving complete remission rates that oncologists had never seen before. This success led to opportunities I never expected. Pharmaceutical companies began recruiting me for research partnerships. The National Cancer Institute invited me to present my findings at their annual symposium. Medical journals were publishing my papers as cover stories. But the breakthrough that changed everything came when I developed a platform technology that could predict drug resistance patterns for any type of cancer, not just the specific types I'd been studying. This system could potentially save the pharmaceutical industry billions of dollars by identifying which drugs would fail before investing in expensive clinical trials. My family watched these developments with a mixture of confusion and concern. They saw me working 100-hour weeks, living in a small apartment near the lab, and having no social life outside of research conferences. What they didn't understand was that I was building something that could save millions of lives. To them, I looked like someone who was obsessed with work, instead of building a real life. They couldn't see that my obsession was actually the foundation for medical breakthroughs that would eventually transform cancer treatment worldwide. But I was too focused on the science to care about their perception, until the day my research finally received the recognition it deserved. The betrayal came from someone I had trusted completely. Dr. Michael Torres had been my mentor at Biogenesis Labs, the senior researcher who seemed genuinely committed to advancing women in pharmaceutical sciences. He was everything I aspired to become: published, respected, and apparently dedicated to supporting innovative research that could change how we treat cancer. It started when Dr. Torres assigned me to lead a groundbreaking project developing personalized immunotherapy treatments. Preparing and narrating this story took us a lot of time, so if you are enjoying it, subscribe to our channel. It means a lot to us. Now back to the story. This was cutting-edge territory in 2021, and pharmaceutical companies were desperately seeking new approaches to harness the immune system's power against cancer. Torres told me this was my opportunity to establish myself as a thought leader in precision medicine. For 8 months, I immersed myself completely in the project. I collaborated with immunologists from Johns Hopkins, analyzed genetic data from over 5,000 cancer patients, and developed a revolutionary approach that could train each patient's immune system to recognize and destroy their specific type of tumor. The work required understanding complex interactions between genetics, immunology, and oncology that few researchers had ever attempted to combine. The breakthrough came when I realized we'd been thinking about immunotherapy all wrong. Instead of trying to boost the immune system generally, my approach identified the exact molecular signatures that each individual tumor used to hide from immune detection. Then I developed personalized vaccines that could teach the immune system to recognize and attack those specific hiding mechanisms. Dr. Torres reviewed my work with obvious enthusiasm. "Rachel, this is revolutionary," he said, studying my research data displayed across multiple screens. "This could make immunotherapy effective for 80% of cancer patients instead of the current 20%. I want to present this to the National Cancer Institute Review Board myself." I was thrilled. Finally, someone would champion my work at the highest levels of cancer research funding. 3 weeks later, I was reviewing patient data when Dr. Lisa Martinez, our lab's research coordinator, approached me with a troubled expression. "Rachel, I need to tell you something, and you're not going to like hearing it," she said quietly. "Dr. Torres just presented a revolutionary immunotherapy approach to the NCI Review Board." My stomach dropped. "That's wonderful. How did the presentation go?" Lisa shook her head slowly. "He presented your entire personalized vaccine system as his own innovation, called it the Torres Protocol. Your name wasn't mentioned once in the presentation materials." The world seemed to tilt. 8 months of my most innovative research, stolen and presented as someone else's breakthrough. I marched to Dr. Torres's office and knocked firmly on his door. "Dr. Torres, I need to discuss the NCI presentation about personalized immunotherapy." He looked up from his computer with complete composure. "Ah, Rachel. Excellent work on the research support for that project. Your data analysis was very thorough." "Research support? Dr. Torres, that was my entire theoretical framework. My approach, my methodology, my breakthrough insight about molecular signatures." His expression remained unchanged. "Rachel, you need to understand how academic research works. Graduate researchers contribute data to senior investigators' research programs. That's how knowledge advances in the scientific community. I refined your preliminary findings, added clinical context, and presented them at a level appropriate for the National Cancer Institute. You stole my research." Torres's demeanor hardened. "I mentored you through a complex project and gave you invaluable research experience. That immunotherapy system wouldn't exist without my guidance and laboratory resources. Besides, who do you think the NCI would rather fund? A postdoc with 3 years of experience or a principal investigator with 20 years of proven research leadership?" I stood there speechless, realizing my mentor had been grooming me not for scientific advancement, but for intellectual exploitation. "I suggest you focus on your next research assignment, and remember that collaboration, not individual recognition, drives scientific progress." That night, I called Grandpa Doc, desperate for support, someone to validate my anger. Instead, he sighed heavily. "Rachel, this is exactly what I was afraid of in academic research. Your ideas are only as protected as your ability to publish them first." The Torres incident wasn't an isolated betrayal. It was the beginning of a systematic pattern that made me question everything I thought I understood about academic pharmaceutical research. Over the next 2 years, I watched the same cycle repeat itself, with devastating consistency across different projects and research collaborations. It started with smaller thefts. My experimental protocols would disappear from shared research databases, only to reappear as collaborative methodologies credited to senior researchers. My innovative drug compound designs were implemented in major studies but attributed to laboratory team efforts, rather than their actual creator. When I raised concerns, I was reminded that scientific progress required selfless collaboration. Then came Dr. Kevin Stone, fresh from Harvard Medical School with an MD PhD and a father who happened to be the chief medical officer at a major pharmaceutical company. Stone was assigned to our research group as my supposed equal, but within weeks it became clear he was being fast-tracked for opportunities I'd been working toward for years. The most blatant example occurred during the Alzheimer's drug development project that could have defined my entire career. I had spent 18 months developing a novel approach to preventing the protein plaques that destroy brain cells in dementia patients. The work required understanding complex neurobiology, designing molecules that could cross the blood-brain barrier, and creating delivery systems that could target specific brain regions. My breakthrough insight was discovering that Alzheimer's plaques formed through a previously unknown inflammatory pathway that could be interrupted using a combination of existing anti-inflammatory drugs with a new compound I'd synthesized. This approach could potentially prevent Alzheimer's progression in early-stage patients, something no current treatment could achieve. But when it came time to present our findings to the Pharmaceutical Research Institute, Dr. Chen chose Stone to lead the presentation. Dr. Stone has excellent clinical background, she explained when I questioned the decision. This requires sophisticated medical knowledge that resonates with physician researchers. I watched from the back of the auditorium as Stone presented my work to the most influential researchers in neuropharmacology. He stumbled through mechanisms he barely understood, mispronounced biochemical terms, and couldn't answer detailed questions about the inflammatory pathways I discovered. Yet when the presentation ended, he was praised for his innovative approach to neurodegenerative disease. The following week, Stone was selected to lead the $15 million clinical trial that would test my Alzheimer's treatment. But the pattern extended beyond Stone. I began documenting how opportunities were distributed throughout the entire pharmaceutical research community. Male researchers with mediocre publication records were selected for high-profile studies. Women with exceptional breakthrough insights were consistently assigned to supportive roles that never led to principal investigator positions. Dr. Jennifer Walsh had developed a revolutionary gene therapy approach that could cure certain inherited diseases. The therapy was patented under her department head's name. Dr. Maria Rodriguez created a new drug delivery system that increased treatment effectiveness by 50%. It became known as the Harrison method, after her male research supervisor. I started protecting my work obsessively. I kept encrypted copies of my original research notes, time-stamped my experimental data, and began filing provisional patents on my innovations before sharing them with anyone. I wasn't being paranoid. I was being strategic. The breaking point came when I was passed over for a senior research position for the third time. My publication record was outstanding, my research was being cited by top journals, and my drug development success rate was higher than anyone in the department. Yet I watched researchers with half my accomplishments and none of my innovative thinking receive promotions to lead major research programs. During my annual review, Dr. Chen delivered the news with clinical detachment. Dr. Kim, you're an excellent researcher, but the review committee felt you need more experience in collaborative leadership. Your recent tendency to assert ownership over research findings concerns senior faculty. I realized my competence had become a liability. Every breakthrough I achieved, every drug I developed, every innovative approach I created, was being used as evidence against me. I was too effective at solving problems, and that made other people uncomfortable. That night, I made a decision that would fundamentally alter my career trajectory. I would stop feeding the system that was consuming my innovations. Instead, I would begin building recognition that couldn't be stolen or reassigned by people who saw my success as their opportunity. The call came at 11 p.m. on a Thursday in October, just as I was finishing data analysis on a promising new cancer compound in my small home laboratory. The voice belonged to my mother, but it carried a weight I'd never heard before. "Rachel, you need to come to the hospital immediately. Grandpa Doc collapsed in his lab tonight. The doctors say he had a massive stroke, and they don't think he has much time." I drove to Stanford Medical Center still wearing my lab clothes, clutching the research journal he'd given me 20 years earlier. By the time I reached the intensive care unit, Grandpa Doc was unconscious but stable, surrounded by machines that monitored every vital function with electronic precision. I sat beside his bed for 4 days, holding his weathered hands and telling him about my research, about the breakthroughs I'd achieved, about the battles I was fighting that had nothing to do with curing diseases. On the 4th evening, his eyes opened with sudden clarity. He couldn't speak due to the breathing tube, but he pointed to the bedside table where a thick envelope sat with my name written in his careful handwriting. Inside was a letter dated just 2 months earlier, along with something that made my breath catch: his original medical school diploma from 1955, and a faded photograph of him receiving a research award for developing an antibiotic that saved thousands of soldiers during the Korean War. But it was the letter that would transform my entire approach to pharmaceutical research. "My dearest Rachel," it began. "If you're reading this, then I'm probably analyzing compounds in whatever laboratory they assign to old scientists in the afterlife. I've been following your career more closely than you know, and I need you to understand something that no one else has had the courage to tell you. You're not just a good researcher, you're a revolutionary one. I've been making calls using connections from my 60 years in medicine and pharmaceutical research, asking questions about your work. Do you know that your cancer resistance protocols are being used by drug companies worldwide? That your immunotherapy approaches have been adopted by research institutions across 3 countries? That pharmaceutical executives are studying your methodologies to guide their billion-dollar research investments? You've been creating medical advances that are changing how the entire industry approaches drug development. But I also know about Torres, about Stone, about all the ways they've tried to diminish your contributions. I've seen this before, granddaughter. During my time, they did it to women researchers, to anyone who didn't fit their narrow definition of who belonged in pharmaceutical leadership. Don't let them convince you that their limitations define your potential. You have something they fear, the ability to see solutions they can't imagine, and the persistence to prove those solutions work. Protect that gift. Document everything. Build your own network. And when the moment comes, and you'll recognize it when it arrives, don't wait for permission to revolutionize medicine." The letter ended with coordinates written in his precise scientific notation. I recognized the format immediately, our old treasure hunting game from childhood, but adapted for laboratory locations. I entered the coordinates and discovered they led to a private research facility in South San Francisco. Inside that facility was a fully equipped laboratory that Grandpa Doc had been secretly funding, complete with state-of-the-art equipment and research materials worth over $2 million. Attached to the lab was an office containing files documenting his correspondence with pharmaceutical executives, FDA officials, and venture capitalists who specialized in medical innovations. At the center of his desk was a folder labeled Rachel's Future. Inside were letters of introduction to CEOs of major pharmaceutical companies, investment proposals for independent drug development, and a research grant application, already approved, for $10 million in funding to establish my own pharmaceutical research institute. Grandpa Doc died peacefully the next morning, just as the sunrise painted the hospital windows gold. At his funeral, pharmaceutical executives I'd never met approached me with stories about his pride in my work and comments about my groundbreaking contributions to medical science. I realized he'd been building a foundation for my independence, while I'd been fighting for recognition in institutions that were never designed to support innovators like me. Standing at his graveside, I made a decision that would define the rest of my career. I would stop trying to earn respect from people who were determined to steal my work. Instead, I would build something so undeniable that they would have no choice but to acknowledge what I'd accomplished. The moment that changed everything happened 3 weeks after Grandpa Doc's funeral, during my sister Emma's engagement party at our family home in Palo Alto. I had just received FDA approval for Phase 3 clinical trials of my breakthrough cancer treatment, the same research that Dr. Torres had tried to steal and present as his own. I also carried the acceptance letter for my appointment to the board of directors at Meridian Pharmaceuticals, the largest biotech company on the West Coast. I walked into the elegant backyard celebration wearing my lab coat, carrying a folder containing patent approvals worth potentially billions of dollars and clinical trial data that could save thousands of lives. The party was filled with 3 generations of family members, Emma's fiancé's successful business associates, and neighbors who measured achievement by traditional markers of success. The conversation buzzed with excitement about Emma's engagement to Michael, a successful venture capitalist, and their plans for a lavish wedding in Napa Valley. Everyone was dressed in cocktail attire, discussing careers, investments, and social connections with the confidence that comes from conventional professional success. When I entered the gathering, conversations gradually quieted as people noticed my unchanged appearance. I looked exactly the same as I had throughout graduate school. Practical clothes, no makeup, hair pulled back for laboratory work, completely focused on research rather than social presentation. My Aunt Patricia, who worked in corporate public relations and had always been the family's authority on appropriate social behavior, looked me up and down with obvious disapproval. "There's Dr. Rachel," she announced to the crowd, her voice carrying that mixture of embarrassment and condescension I'd heard my entire life. "Still playing scientist instead of getting a real life. You're embarrassing Emma on her special day, so please leave." The party fell silent. I could feel every eye on me, see the uncomfortable expressions from relatives who agreed with her assessment. But something inside me shifted. Not into hurt, but into absolute clarity. "Actually, Aunt Patricia, I need to share something with all of you." I pulled out my phone and opened the email that had arrived that morning from the FDA. "This morning, I received approval for Phase 3 clinical trials of a cancer treatment I developed. Apparently, the Food and Drug Administration believes my research could save 50,000 lives annually." The gathering went dead quiet. I could see my father's confused expression, my mother's dawning realization that she didn't understand what I actually accomplished in my laboratory work. But I wasn't finished. I opened my laptop and pulled up the presentation I'd been preparing for my board appointment. "Since we're all here, let me show you what playing scientist actually means." For the next 20 minutes, I walked them through my pharmaceutical achievements. The cancer treatments now in clinical trials at major medical centers. The drug resistance protocols being used by pharmaceutical companies worldwide. The patents I held on compounds that could revolutionize how we treat genetic diseases. I showed them screenshots of emails from pharmaceutical CEOs, letters from the National Institutes of Health, and articles from medical journals featuring my research breakthroughs. I displayed my patent portfolio valued at over $500 million, my consulting agreements with biotech companies, and the board appointment that made me one of the youngest pharmaceutical executives in the industry. When I finished, my Uncle Robert, who had always been the family's most scientifically minded member, broke the silence. "Rachel, are you telling us you're some kind of pharmaceutical executive that major drug companies actually rely on your research?" I looked directly at Patricia. "I don't just work for pharmaceutical companies, I help run them. While you've been worried about my social presentation, I've been developing treatments that will be prescribed by doctors worldwide." The party remained silent as I packed up my laptop. "So you're right about one thing, Patricia. I'm not living a normal life. Normal researchers work on incremental improvements. I develop breakthrough treatments that change medicine." I looked around the gathering one final time. "Congratulations to Emma and Michael. I hope you understand now that success isn't always measured by social conventions." 2 days later, my phone rang with a call that would validate everything I'd built. "Dr. Rachel Kim, this is Jonathan Sterling from Meridian Pharmaceuticals. We've been following your cancer research breakthroughs, and we'd like to offer you a position on our board of directors to oversee our oncology division. Can you meet us at our headquarters tomorrow?" The call should have been the ultimate validation of my research career. Instead, it became the beginning of the most challenging period of my professional life. Because operating at the highest levels of pharmaceutical leadership comes with powerful enemies who want to see you fail. The moment news of my board appointment reached the academic research community, the retaliation began swift and systematic. Dr. Torres, who had spent years appropriating my work, suddenly became very interested in my business activities. "Rachel, I've been hearing concerning things about your transition to corporate pharmaceutical work," he said when he called me the day after the Sterling meeting. "Board positions and patent ownership? This level of commercial involvement raises serious questions about conflicts of interest and research integrity." "Dr. Torres, my pharmaceutical work is fully disclosed and ethically structured through proper technology transfer channels." "Technology transfer of research developed using university resources," he said, his tone growing cold. "I've been consulting with the ethics committee, Rachel. Your rapid move into commercial pharmaceutical leadership is creating concerns about exploitation of academic research for personal profit." I realized he was building a case to undermine my credibility. Every patent I'd filed, every consulting agreement I'd signed, every pharmaceutical partnership I'd developed, it was all being reframed as evidence of unethical behavior and academic misconduct. Over the next month, my life became a gauntlet of professional sabotage. Torres began spreading rumors about my undisclosed commercial interests and abandonment of scientific principles. Other researchers who had previously been collegial started questioning my motives and suggesting I was exploiting academic resources for personal gain. The most devastating blow came when Dr. Kevin Stone, leveraging his father's pharmaceutical connections, began interfering with my clinical trials. Research hospitals I'd worked with for years suddenly raised concerns about my commercial bias. FDA reviewers who had previously fast-tracked my applications began requesting additional safety data that would delay trials by months. I spent sleepless nights managing regulatory challenges while trying to advance life-saving treatments through bureaucratic obstacles. But the real challenge wasn't the operational setbacks, it was the isolation. Academic colleagues who had previously been supportive became distant. Medical conferences where I'd once been welcomed now felt hostile. I was being systematically excluded from the networks that make pharmaceutical research possible. The breakthrough came from an unexpected source. Dr. Jennifer Walsh, the researcher whose gene therapy had been stolen and credited to her department head, approached me at a biotech industry meeting in San Francisco. "Rachel, I know what they're doing to you," she said quietly. "The same thing they did to me, just not as publicly. But I want you to know, there are others of us watching. Women researchers who've had our discoveries appropriated, our commercial opportunities blocked, our transitions to industry sabotaged." She handed me a secure communication device. "These are contacts across the pharmaceutical industry who've experienced similar treatment. We've been documenting patterns, sharing resources, building our own research and development network." That device contained contact information for 37 women pharmaceutical professionals across different companies and specialties. All of them had experienced systematic undermining of their career advancement. All of them had been fighting the same battles I was fighting, individually and in silence. For the first time since the Sterling call, I didn't feel alone. "Jennifer, what are you suggesting?" "I'm suggesting that maybe it's time we stopped competing for acceptance from people who are determined to exclude us. You're operating at a level none of us have ever reached. If you succeed, if you prove that innovation and results matter more than politics, it changes everything for all of us. And if I fail, then we'll still be here, ready to support the next woman who breaks through." I looked at those 37 names and realized I wasn't just building a pharmaceutical career. I was carrying the hopes of every woman researcher who'd been told she wasn't business-minded enough, leadership-ready enough, or commercially sophisticated enough for pharmaceutical executive positions. The Meridian Pharmaceuticals boardroom felt like the epicenter of medical innovation as I took my seat among the most influential executives in the biotechnology industry. 12 board members representing $200 billion in pharmaceutical assets, with the power to determine which treatments would reach millions of patients worldwide. Dr. Kevin Stone sat 3 seats away from me, nervously reviewing notes about drug development projects he'd never actually managed. But I was no longer concerned about his presence threatening my position. I had something more powerful than his family connections. I had breakthrough treatments that could revolutionize medicine. The meeting began with CEO Jonathan Sterling asking a direct question. "Dr. Kim, our oncology division is facing a crisis. 3 major cancer drugs have failed in late-stage trials, costing us $2.8 billion. How do we prevent this from happening again?" For the next hour, I dismantled conventional wisdom about pharmaceutical development. I explained how traditional drug testing relied on outdated models that couldn't predict real-world treatment outcomes. I demonstrated how my resistance prediction protocols could identify drug failures before companies invested hundreds of millions in doomed clinical trials. But the moment that changed everything came when Sterling pulled up financial projections showing potential losses from our current drug pipeline and asked me to recommend which projects should be continued or canceled. I walked to the presentation screen and analyzed each of our 15 drugs in development. This Alzheimer's treatment will fail because it targets the wrong inflammatory pathway. This diabetes drug will show initial promise but lose effectiveness after 6 months due to cellular adaptation. However, this overlooked compound for rare genetic diseases could generate $5 billion annually if we modify the delivery mechanism. The room went completely silent as I outlined specific modifications for each drug, including molecular adjustments, patient targeting strategies, and regulatory pathways that could accelerate approval timelines while reducing development costs. Sterling leaned forward. Dr. Kim, you're describing modifications that could save our company $2 billion while developing treatments that could help millions of patients. How confident are you in these recommendations? Confident enough to stake my board position on their success, I replied. Implement my protocols, and I guarantee we'll have the highest drug approval rate in the industry. By the end of the meeting, I had secured approval for $500 million in research modifications based on my analysis. More importantly, I had gained operational control over our entire drug development pipeline, something no board member had ever achieved in their first year. The validation came immediately. Within 6 months, every drug modification I'd recommended proved successful in clinical trials. The Alzheimer's treatment that would have failed was redirected to become a breakthrough therapy for early-stage dementia. The diabetes drug was reformulated to provide sustained effectiveness. Most dramatically, the rare disease compound became our most profitable treatment. But the real breakthrough was systematic. Meridian's drug approval rate increased from 12% to 67% under my oversight. Other pharmaceutical companies began requesting consultation on their failed drug programs. The FDA started using my resistance prediction protocols to guide their approval processes. The network of women researchers that Jennifer had connected me with began receiving executive positions through my recommendations. For the first time in pharmaceutical history, leadership decisions were being made based on scientific innovation rather than corporate politics. 8 months later, when Pharmaceutical Executive magazine published their annual list of industry leaders, I was featured on the cover as the researcher who revolutionized drug development. The most satisfying moment came when Dr. Torres requested a meeting to discuss collaboration opportunities. He wanted access to my drug development insights and board-level connections, the same research he'd previously stolen and minimized. I politely declined but offered to recommend some of the women researchers from my network who were seeking leadership opportunities with experienced pharmaceutical executives. The girl who had been told she was embarrassing the family by focusing on research was now setting the standard for how the pharmaceutical industry approached drug development and clinical trials. But more importantly, I had proven that scientific excellence and innovative thinking matter more than academic politics and social expectations, opening doors for every researcher who had been told they didn't belong in pharmaceutical leadership. The call came 1 year after my appointment to Meridian's board had transformed me into one of the pharmaceutical industry's most influential leaders. The voice on the other end was familiar but carried a humility I'd never heard before. It was my father. Rachel, your mother and I have been talking about what happened at Emma's engagement party, about everything we said. We think we owe you more than an apology. His voice broke slightly. We'd like to take you to dinner. Somewhere special. There are things we need to understand about what you've accomplished, about who you've become. 3 days later, I met them at the French Laundry in Napa Valley, a restaurant that represented the kind of success they finally understood I'd achieved. Dad looked older, more uncertain of himself. Mom walked beside him carrying a folder against her chest like armor. But it was what Dad wore that surprised me most. A small pin with the Stanford Medical School logo, as if he'd finally understood that my education had led to something extraordinary. We were seated at a table overlooking the vineyard, the same landscape where pharmaceutical executives and biotech investors often held their most important meetings. Dad looked around at the elegant atmosphere, the sophisticated clientele, the understated luxury that I now navigated as naturally as breathing. Rachel, he began, his voice unsteady, I owe you an apology that should have come years ago. Mom opened her folder and pulled out printed articles, medical journal publications, and pharmaceutical industry features. After the engagement party, we realized we had no idea what you actually do, she said quietly, so we started researching. We talked to people in biotechnology. We learned about your board position. Dad picked up a copy of Nature Medicine with my photograph on the cover. I learned that you're not just a researcher, Rachel. You're considered one of the most innovative pharmaceutical executives in the country. That your treatments are saving lives at major medical centers. I also learned something else, he continued, meeting my eyes for the first time in years. I learned that your pharmaceutical patents and board compensation far exceed anything I ever earned in my entire software engineering career. Despite everything, I almost smiled. Of course, Dad would finally understand success in financial terms. But that's not why I'm here apologizing, he said quickly. I'm here because I finally understand what I was too conventional to see. You weren't avoiding real life. You were building something I was too limited to comprehend. Mom leaned forward. Rachel, we attended a medical research fundraiser last month. They had a guest speaker who talked about women in pharmaceutical leadership. She specifically mentioned your cancer treatments, called you, quote, the researcher who's changing how we cure disease. We sat there realizing our daughter was the person she was praising, and we'd spent years making you feel ashamed of your dedication. The conversation that followed was the most honest we'd ever had. Dad admitted that his criticism of my career focus had been rooted in fear that he couldn't understand my world, fear that I was sacrificing social connections for professional obsession, and fear that my unconventional path highlighted the limitations of his traditional thinking. I spent so many years trying to convince you to choose work-life balance over scientific excellence, he said. I never considered that maybe your definition of balance was different from mine, that your life's work was literally saving lives. Mom pulled out one final document from her folder, an invitation addressed to our family. This came to our house last week, she said. It's from the American Medical Association, inviting us to a ceremony where you're receiving some kind of lifetime achievement award. I looked at the invitation. I hadn't told them about the ceremony because I'd assumed they wouldn't understand its significance. We'd like to attend, Dad said, if you'll have us. And Rachel, I want to learn about your research. Not because I finally approve, but because I want to understand the daughter I should have supported all along. For the first time in my life, when Dad introduced me to the restaurant staff and other diners, it wasn't with embarrassment or confusion. It was with the kind of pride that comes from finally recognizing achievement, even when it appears in forms you never expected to understand. 2 years later, I stood at the podium of the largest pharmaceutical conference in the world, looking out at an audience that represented the future of medical innovation. The occasion was the launch of the Kim Institute for Women in Pharmaceutical Sciences, my new foundation dedicated to developing the next generation of women researchers and executives. In the front row sat my parents, both wearing their finest clothes and beaming with a pride that had taken years to develop. Next to them was Dr. Jennifer Walsh, now my chief research officer and one of the most respected pharmaceutical executives in the industry. Behind them were dozens of women researchers whose careers had flourished under the mentorship and funding programs we'd established together. But it was the young woman in the third row who captured my attention most completely. Dr. Sofia Martinez, a recent PhD graduate with groundbreaking research in gene therapy and dreams of developing treatments for rare childhood diseases, held a small notebook and absorbed every word with the same intensity I'd shown 20 years earlier. She reminded me of myself at that age, brilliant, determined, and completely unaware of the obstacles she'd face simply because she was a woman in pharmaceutical leadership. 3 years ago, I began my address to the assembled researchers. I sat at a family engagement party and was told I was embarrassing my sister by focusing on research instead of building a real life. Not because I had failed, but because the people who loved me most couldn't understand that some callings require everything you have to give. I paused, scanning the faces before me. Today I want to talk about what happens when you refuse to let other people's limited vision define your potential to heal the world. I told them about Grandpa Doc's research journal, which now sat in a place of honor in my office next to photographs of me with FDA officials, pharmaceutical CEOs, and the keys to laboratories developing treatments for diseases that were considered incurable just 5 years ago. I spoke about the network of women researchers who had supported each other through systematic exclusion and professional sabotage. I explained how documenting our breakthroughs and building independent recognition had created opportunities the traditional system would never have provided. But the real victory, I continued, isn't my board positions at 3 major pharmaceutical companies, or my portfolio of patents worth over $800 million, or even the Presidential Medal for Scientific Excellence I received last month for developing breakthrough cancer treatments. I looked directly at Dr. Martinez. The real victory is that tomorrow a young woman will enter a pharmaceutical laboratory in a world where her innovations won't be stolen, where her research will be valued, and where her path to developing life-saving treatments won't require her to fight the same battles we fought. After the presentation, Sophia approached me with nervous energy and determined eyes. Dr. Kim, I've been developing some theories about using CRISPR technology to correct genetic mutations in real time within living patients. My advisors say it's too ambitious for early career research, but after hearing your story, I smiled, remembering my own desperate attempts to share innovative ideas with researchers who saw potential as a threat. Sophia, ambitious thinking is exactly what we need to cure diseases that have never been cured. Let's discuss it. Over the next hour, she outlined a strategy for using gene editing technology to develop personalized treatments for over 200 rare genetic diseases. Her thinking was sophisticated, original, and exactly the kind of innovation that would define the next generation of pharmaceutical breakthroughs. Sophia, I'm offering you a $2 million research grant and a position as principal investigator at the Kim Institute. Your CRISPR research will be developed under your name and credited to you. 6 months later, Sophia's gene therapy approach was being tested in clinical trials at major medical centers. She had become the youngest researcher to receive FDA fast-track approval for a breakthrough therapy designation. The story that began with a compass and a grandfather's faith in scientific discovery had become something larger than either of us imagined. It became proof that when you refuse to accept limitations others place on you, you don't just change your own career, you create new possibilities for healing that benefit everyone. Every time a young woman receives funding for groundbreaking research, every time innovation is credited to its actual creator, every time the system chooses scientific excellence over politics, Grandpa Doc's journal continues pointing toward true medicine. That direction is always toward healing. Up next, you've got 2 more standout stories right on your screen. If this one hit the mark, you won't want to pass these up. Just click and check them out. And don't forget to subscribe and turn on the notification bell so you don't miss any upload from us.